going to be about pharmaceutical manufacture. So we're going to take a break from the hard science, like Western blot, and we're going to and like different adrenergic drugs and all that stuff. We're going to look at pharmaceuticals from a processing point of view. Yes, we understand that these drugs take years to discover, but once they're discovered, what's the good of it if we can't produce them in mass scales, right? So we want to go from the early stage drug discovery where we have a lead candidate and we know exactly what form to put it in, either oral, by a tablet, by a liquid, by an injection, by whatever it may be. And we want to put that into a large-scale manufacturing protocol that follows all of the FDA regulations. We want to make it so that we don't lose material, that it's cheap as can be, which is usually very expensive, and that it produces good quality drugs. We need QCing is one of the most important parts of manufacturing. So let's take a look at the overall process of drug manufacturing. So first, we went over this with the whole drug discovery workflow, where we identify the drug targets. So we're going to use all of our assays, all of our technology to do that. We optimize. So that's really all we talked about in this class. Today's lecture is going to be on the rest, more the middle part. So then, after we figure out our drug, we optimize manufacturing to reduce costs. So there's an entire team that streamlines the manufacturing process, consisting of project managers, manufacturing people, and entire teams of manufacturers, and different equipment managers, people that are specifically designated or consultants for one piece of equipment. Maybe it'll be a compressor that compresses the pills together. So you really coordinate with many teams of people, and sometimes at different sites, to have the process streamlined enough to reduce costs and limit emission of toxic chemicals, etc. Then you implement batch manufacturing processes. This means we take a large-scale synthesis of the drug. So let's say we're talking about a small molecule drug, which most drugs are. 90% of drugs are small molecules. We are going to synthesize this small organic molecule on a batch basis. You take a large vat of hydrochloric acid. It mixes with something else, and it mixes with a byproduct, and then you mix it and do your different reactions that you would on a benchtop, but in large chemical factories. Then you design, which is still, this part is still tough to do, the transition from implementing a batch manufacturing process, which is batch to batch, versus a continuous manufacturing platform. So continuous manufacturing platform, that means a assembly line of steps of this synthesis, that one part is not waiting for the previous part. It is consistent and it doesn't stop. This is very difficult to do because most drugs in current times are synthesized and produced using batch manufacturing. Continuous manufacturing is better, and we'll see slides on why this is, but there's a lot of hurdles in transitioning in the batch manufacturing of Advil and completely changing that manufacturing style, even if it's better for the environment, it saves money, and it saves time. But they've been producing Advil for a really long time using batch. So we're just going to leave it that way because it works. That's what these higher ups decide, which is good for now, but maybe when, let's say, we're at a population of 9 billion, that might not be the best thing anymore. So anyway, the next step, which is the future, that's why I have the future question mark, is create small personal manufacturing platforms. Now, this part is more theoretical than ever because drugs right now are manufactured in large chemical plants. There really isn't any way around that. The goal is to get drugs to places on Earth that cannot have access to drugs readily. So let's say you have remote parts of Asia, Africa, South America, even Canada. I mean, there's people that live like in little villages in Canada that don't have access to drugs. They need to drive 5 hours to go to a supermarket or a pharmacy. right? And Europe too, and Russia, right? There's like all these huge places that are that don't have access to medication. And if we made small scale manufacturing platforms that are self-sustaining to make common drugs, or even to interchange between making different types of drugs, that is a goal for overall healthcare that will save a lot of lives. So it's one thing, and they're equally important. I mean, me coming from the identifying target drug department, I think that's more important, but the manufacturing is equally as important. Because if you can manufacture and distribute to a wide range of people and a lot of different places, that is as important as the discovery in the first place. And we saw that firsthand during the COVID vaccine. Distribution was very good. We got it out it was made in, let's say, December of 2020, it was finalized. January, it started coming out to niche populations. Like I didn't get it until later, but I had access to get it in January because I was a professor. But I mean, I didn't want to get it because of I wanted to let the healthcare workers get it first. I think that's more important. I mean, what I do here is, and at that time we were still virtual, so I wasn't really at risk anyway. So I just wanted the people. I didn't want to take a dose away from either someone who's elderly, at risk, or a nurse or a doctor. So anyway, but the distribution was very good in that process. Right. Right. So there was a lot of countries that had restricted access. So that was a big hurdle, and that's something that we're constantly trying to mitigate. So anyway, so here's another more detailed example of the workflow. So you have of when you already streamline the process. Here's what we're going to talk about today. So you have material, you get the material, you get a reactor. So when we think of the word reactor, we think of nuclear reactor, right? No. We're talking about basically a large test tube, a large beaker, right? We are mixing the reactants together and we're making products. So a large chemical reactor. So it assesses reaction completeness, impurities, kinetics, and solids. So that's a lot of factors that go into it. Then the workup. So the workup means we're measuring the yields. We're taking the next step and we're assessing the impurities using a lot of analytical techniques, mass spec, IR, different types of chromatography. And mass spec is part of all of this, but it's not listed here. I'm not sure why. But it's listed, it says here, MS equals mass spec, but it's not listed under any of these. Oh, here it is. But it's really should be under all of them. So then crystallization. So depending on the type of drug, right? Some drugs we may want to be soluble in a liquid suspension. Some drugs you might want to be in like an emulsion, like a lotion. So some lotions are drugs. They have active ingredients in them. Like different acne lotions. They have benzoyl peroxide, right? Or and a bunch of other stuff. So that's an active drug. But the drug in that case is in an emulsion. It's not in a tablet. It's not in a liquid, a non-viscous liquid like water, right? So the crystallization or whatever form it needs to be in, you work on that. Then if it is crystallization, there's always going to be moisture involved. So whenever you crystallize something to make it a powder form, there is moisture and there's solvent content that needs to be eliminated. So then you can go through filtration, drying, and work on that. Then milling. So milling is kind of separating, is a funny word for separating and mixing. So you separate and break down the crystals to make a consistent powder. Then the consistent powder can go into tablets. And we'll talk about exactly how those tablets are made. There are 3 different ways tablets can be made. And we'll talk about those today. And across every aspect of this workflow, we are introducing analytical techniques to QC every single step. So this is more of a batch. This is more of a batch orientation, and continuous manufacturing will kind of morph the same processes into a continuous workflow that the materials are always there, and you don't wait for one process to finish before the other one. So they're all happening at the same time. And then it's just like a constant conveyor belt, which is very difficult to do. Because if, let's say, crystallization stops, that means everything else prior is stopped. In a batch, if crystallization is stopped, let's say there's an impurity in the crystallization that's not supposed to be there, you can take it out, just like get that batch, just move it aside, and start with a new that'll delay the time, but it will, in continuous, that'll create more of a hang up. Yes. Yes. So margins of error, I'm not sure, but contamination they are. So Daniel asked, for those of you online, he said that he asked, is continuous manufacturing more prone to errors, right, and impurities, contamination, and impurities and stuff? So yes, it is, and also it has more waste involved. So there's a lot, there's more moving parts, more complicated, more chance for error. That's the basic gist of it. Okay. So let's take a look into that more in detail, unless there's any other questions. Are there any questions on that? Okay. So batch versus continuous. So batch, we have various components are brought in the simple step-by-step process, and then the current batch is finished before the new one begins. So most drugs are performed or manufactured in batch settings. So you take the raw material, you do everything you need to do to it, and then the product comes out. And I'm being simple right now because I wasn't simple before, and then we'll talk more in detail later. So just batch. Batch is the simple one. Continuous, it combines the full manufacturing workflow stream into a single integrated flow. This means that all of the processes are happening at once. If you can see from the schematic on the right, bottom right, raw material comes in, product. Then raw material comes in, another product is formed, and it kind of goes step by step. So it's one product, the first product directly goes into the second reaction, which then directly goes into the third, and it keeps, and then behind each one, there's a previous reaction happening, and it keeps going. It's faster. It's theoretically a lot more efficient. So let's see. So it eliminates the production gaps, shortens manufacturing time, saves money, and of course generates more revenue because you can push out more drugs. It's very new, and the more drugs that are being developed, the more that they're pushing towards continuous. But there's a lot of hurdles. One, it requires a huge investment, and it requires a lot of time to convert already established batch techniques to continuous techniques, which it is not in the favor of a lot of different pharmaceuticals, especially in America, because for common drugs, there's never really a shortage of them. So like, have you ever seen an Advil shortage? No. Or like different like multivitamins. There's never really a multivitamin shortage, right? They all have their batch production down to a T. So they don't really have a reason to change it. So advantages and disadvantages. We kind of went over this a little bit verbally, but let's just take a look. So batch manufacturing, they're well established. There's batch manufacturing techniques that are virtually unchanged since the 70s or 80s. Why? Because they work, and because they produce the revenue that the company gets, or that the company, that the company likes. Yeah. So also they're easier to approve. So there's something that we're not going to talk about today are FDA and manufacturing regulations, government regulations on drug manufacturing. There are so many that we don't have time to talk about them. And also, depending on if you go into this industry, there are so many different ones that you'll learn which one you need to worry about, because each step requires a different type of regulation. So we're not really going to cover all of them, but there's a lot of regulations. So batch manufacturing, because it's more simple, getting the approval for the regulations, or getting the approval is easier. So sometimes it may be the only viable method to produce some drugs. So for example, what if a drug is very sticky and you can't use the same vat without cleaning it in between batches? Well, then you wouldn't be able to use a continuous workflow, because then over time, the batch that is mixed in that vat that gets dirtier and dirtier and dirtier, the quality will decrease over time, and there may be more impurities. So if someone takes a drug that is earlier in the production line versus later in the production line, they're getting a different drug. There's different impurities, and there's maybe more side effects. So this is why sometimes it's not in the best interest to use a continuous method for already established batches. Also, there's low waste. So you use the batch, you take in all the raw materials, you make the stuff, there's some byproducts, but then there's no extraneous waste. Disadvantages: it's lengthy. It's longer than continuous. There's hold times might increase because if one thing stops, then the whole batch is maybe lost. There could be bad batches. Sometimes you might have multiple sites for each process. And what that means is there may be one company in China that does part A of reaction A. Then it needs to be sent to India for reaction 2. Then it might need to be sent to Croatia for reaction 3, right? That takes a lot of money, a lot of customs and shipping and time, and a lot of manpower to do. So that's not in the best interest of output. Also, you need more manual QC and manual monitoring. So continuous process requires a lot less FTEs, so full-time employees, than batch manufacturing. Batch requires a lot more people, so hence making it cheaper or making it more expensive in the long run. Continuous. So continuous advantages: reducing the cost, shorter production time, reduces the risk of human error if you have a really good streamlined process, improved quality, blah, blah, blah. It's really good. That's basically the point. It's the best one. But the disadvantages in some cases outweigh the advantage. Is that we already have the established batch technique. The continuous manufacturing requires a higher initial cost. The reason is because the instrumentation and the equipment we're using is very high end. It's very state-of-the-art equipment. This equipment is, one, expensive, difficult to maintain, and let's be real, there may not be employees that know how to use it. So that's training, that's getting different consultants involved, different companies involved to run this equipment, right? So it may be just more difficult for the company to undertake, and they might not want to do it. There's more waste because you're having a constant inflow of products, so then of reagents. So there's going to be more byproduct and more waste. Can't really give a better explanation than that. It's just more waste. So think about it this way: if you have a continuous sheet of solid pills, that the pills need to be stamped out of a sheet, or a tablet needs to be stamped out of a sheet. Imagine what creates more waste: if you make a batch of sheets and then just like cut them all up, or if you take a continuous sheet and like stamp them out. A continuous sheet will probably create more waste over time. But there might be breaks in the sheet, or like it's not going to be fully continuous. There's going to be overall there's more waste. I'll leave it at that. I'll cut that part out. Cut it. So anyway, if there's here's where it's actually bad. If there's a fault in the process, the entire production line stops. So this is a big reason why pharmaceutical manufacturers are scared. It's the 8 o'clock. What is it? 7:30. Are scared to go to continuous manufacturing because if there's one mistake in this very new process, it can shut down the entire line for a couple of days maybe, and that's a lot of revenue that they're losing. So they might want to just stick with what they know. Okay, any questions on this? Okay. All right, so let's talk about different drug classes. So we've talked about these a little bit already, but there's tablets. There's a lot of different types of tablets. Oral tablets, which are the main ones, which are pills. There's chewables, like the gummy vitamins. There's Buccal or sublingual. So like what was it? What was the Icebreakers liquid ice? That's one, right? Remember that one? That commercial? Icebreakers liquid ice. Do you remember that commercial? It's like this. It was a mint that you put it under your tongue and it dissolves over time. And it just makes your breath smell better, and it's time released. So that's an example of like a sublingual. It dissolves over time. Lozenges, so like cough drops, stuff like that. Enteric coating. This is important. So enteric coating, which we've seen on a lot of drugs, is a capsule. So there's a difference between a normal tablet, which is kind of rigid and rough, versus the smooth tablets that look like they have plastic over them. Those are what we call an enteric coating. And the reason why, do I even write it? Why? No, I don't write it. So the reason why, and this is important for a homework question, by the way, the reason why we have an enteric coating, and can anyone guess? Or does anyone know what an enteric coating is? Well, I just told you, it's like it looks like when a pill is laminated. Yeah, a lot of vitamins are like that. Do you know why? That's one aspect. It's easier to go down, yeah, but more importantly, something else. You know?
Does it help it get where it needs to be in the body? For example, maybe the coating might not break down as easily in the stomach if you want it to get to your intestines and break down.
Yes, Adrian, but 1 second. One of your classmates was talking. I'm sorry, you didn't know that because of the disconnect. But it's okay. Good answer, Adrian. And what were you? Yes. So you're both perfectly right. So that's exactly the right answer. So the reason is because we don't want the drug to react with the stomach acid. And also, the reason why we don't want the drug to react with the stomach acid, like Adrian was saying, is maybe the drug is absorbed in the small intestine or the colon, right? In that case, we want the drug to bypass the stomach, and we want it to be protected. That's why we use an enteric coating, to protect the drug from the stomach acids. Good job, guys. All right, and then we have sustained release tablets, well, like liquid gels and stuff, whatever. Okay. Anyway, so yes. So extended release. I think they overlap. So is there a difference? I don't think there's much of it. I think they're just different names. Let me see. Sustained release versus extended release. So there's so they're the same thing, but there is an immediate release. So sustained release and extended release is the same thing. Right. But I need to double check myself. And then there's immediate release, which is immediate release. So that means you like you take the drug, it immediately gets absorbed and then goes into your circulation. So, but then there's sustained release, which means that the way the pill is designed is that it breaks down over time, and then it releases it. The drug is absorbed over the course of maybe 6 or 12 hours. And extended release is the same thing, but extended release is when they say a drug is extended release, it's probably longer. It's a really, let's say, sustained release, but on steroids. It's really strong. Yeah, it's maintained sustained release. That's a good way to say it. Okay, good question. Yeah, very good questions, because I might not know the answers to every single question with manufacturing because it's not my field, but I do know the basics. So anyway, so important factors in making any tablet is right. Like, how do you make a tablet? So we're going to learn about the different types of granulations, but it's not just the active drug. Most of the tablet is not the active drug. We have the inactive ingredients. We also have sometimes solvents when we have wet granulation. We have that's really it. But we have a lot of other things too. We have different types of active ingredients. Also binding agents, which are technically an inactive ingredient, but they help bind the powder together. Also, storage is important. Depending on what type of tablet you have, the storage may be different. If it's a liquid, usually the storage is in refrigerator or freezer temperatures. If we have a biologic, like an antibody or a protein or a peptide or vaccine, in the freezer usually. So and things to keep in mind is like if you're in the manufacturing pipeline, it's literally clogging pipes. So all these pipes that the material goes through, the powder material, or especially with wet granulation where there's liquid involved, you need to have agitators. Every pipe has an agitator on it in order to release all of the gunk that gets built up on the walls, kind of like a blood clot or a clot. We're kind of releasing that. And of course, with every drug manufacturing process, we need to keep an eye on impurities. And also homogeneity is important, and then the extent of mixing. Those are very important things. Okay, so then another type of drug that was tablets. Now we're talking about liquids. So there's water-based liquids, which are most of them, which are really all of them. There's solutions, syrups, lotions, suspensions, which are lotions or emulsions. And usually liquids have a more complex manufacturing and also require more storage or more strict storage conditions, and they usually degrade faster. This is why they're usually more expensive. So you can have a liquid version of most over-the-counter or most prescription tablets or over-the-counter tablets. So important factors for them: storage, the tank selection. We'll talk about that in detail. Long-term storage, purity, API. So API stands for active pharmaceutical ingredient, just to let you know. And inactive binding solubility, solution solubility, solvent, and machinery, right? So those are all things to keep in mind when you're doing this, if you're doing this. And then the last class is biologics. So biologics are the most expensive to make, and the vaccine is under a biologic because it's RNA-based. So are monoclonal antibodies, peptides, different types of gene therapy, and other drug chemotherapeutics. A lot of times can be antibodies, so they go under this. So big factors you have to worry about are immunogenicity, meaning is your immune system going to attack this antibody or protein? Also, different adverse side effects, because you're going to have mechanistic side effects. If you're treating your body with RNA or with a protein, you might have biological reactions in your cells to that protein. That protein might do something and then cause a different cellular response. So also you have to keep in mind pH, media, aggregates, media meaning cell media, so cell culture media. And a lot of these biologics are derived from biologics, meaning cells, meaning things that are alive. So you need to have huge cell culture. We were just talking about cell culture in a petri dish, right, with Western blot. This is going to be huge scale cell culture and taking out all of the antibodies and hyper expressing these cell lines with antibodies or peptides. Anyway, so then storage is very important there too. Okay. Any questions? No? Cool. Next, so we'll talk about different tablet manufacturing processes. So, but do you know these well? Okay, good, awesome. So you can probably help me. So then, so we have three. So there's direct compression, otherwise known as direct granulation. There's wet granulation and dry granulation. So what is granulation to begin with? So it's the process of collecting particles together, creating bonds, and then compressing them together either by themselves or if you need a binding agent to do so. And this is what makes the pill, a pill. And it's the important things to know is that we want to avoid heterogeneity. We want all of the, which is difficult. So if we have a tablet that has multiple solid components that we're mixing together, you can imagine, unless we have to mix them very, very well, more mixing than we would for liquids. Because if we mix solids, if they're a little bit heterogeneous, one of the tablets might be stronger than the other. We can't have that. That goes against regulation. Every pill needs to be well, not every pill, but every batch needs to be QC to make sure that you take like 10 random pills in the batch, that they're all the same composition. Also, impurities. So impurities are more frequent in granulation techniques. And there's many reasons for this, but one reason I think is very interesting is that if you have a manufacturing plant that's working on 3 drugs at the same time, and they're all tablets, you're going to have a lot of dust. And this dust might contaminate another drug that's on the other assembly line. See, the impurities might be due to another drug. And that's not true with liquid formulations because it's all in closed off pipes and there's no dust with liquids. So yeah, and also we need optimized compaction characteristics. So like the different forces involved, the instrumentation, the equipment involved with the compaction. So here's dry granulation. So this is a lot of information on it. We're not going to need to know all this information. We'll just need to know what it is, maybe one pro and one con. That's it. So in dry granulation, components are mixed and they're mixed and they're compressed, mixed and compressed into larger tablets. So it's mainly used for high dosages. So like if you have a multivitamins are pretty big, and usually they're made by dry granulation. Also, probiotics are pretty big too. They're made by dry granulation. So also moisture sensitive drugs like aspirin, where you can't have any solvents involved. Also, just drugs that, after trial and error, don't compress well with wet granulation. Dry granulation might be the best option. So on the right, you see equipment. So there's two ways of granulation. There's one, slugging, which is you take a press and you just take after mixing the products, after mixing the reagents, you press it with a lot of force. Then there's roller compaction. So roller compaction is, you can kind of see by the instrument right here, there's multiple rollers. It pulls it with high pressures, eventually you have or tablet. So those are both ways of granulation, of dry granulation. The positives, so the advantages of dry granulation. The equipment is rather small. It's cheaper on average. It's easier to clean. And they can be used in both batch and continuous production if made correctly. So let's say if the roller if either one of these was hooked up to the previous reaction, the previous mixing, and that mixing went directly into here, and you had connected the instruments together, then you can make a continuous process. The negatives, so no uniform color distribution. It's a small negative, but ideally you want to have your drug to have uniform color. The reason why is to make the patient feel better. Because if a patient has a drug where it's like, hey, one half of this is white and the other half isn't, and you look at another pill, this one looks a little different. Am I taking the right drug? Is it homogeneous? It might be homogeneous, but the color might not be. So that's a negative. Also, there's dust. There's dry granulation. There's cross-contamination with other drugs, like I mentioned. So any questions on dry granulation? No. I would just say know what it is compared to wet and compared to direct. Okay. So here's wet. So wet granulation is similar. You're going to have the same steps, but you're going to have wetting and drying processes. So as you can see from the top right, we're drawing up a comparison between all three techniques. So wet and dry have really all overlaps, except for wet has a wetting process, milling, and drying. Dry also has milling, but it doesn't have 2 stages of milling, and it also has compressing. Wet granulation, you don't really need to compress as much. It requires a lot less force. So anyway, the way you do wet granulation is you take the components, you mix them together with solvents or liquid adhesive, and that binds the powders together. And then you mill it, and then you dry it down, and you mill it. So you mix it up and kind of try to refine it. Then you do it again, meaning you wet it again, and you mix it again, and you get bigger and bigger particles as you keep going through this process. And then eventually you reach the consolidation of a larger particle. Yeah, so some positives is you're going to have more of a consistent distribution of color, and it's easier in general to make homogeneity possible for the drug because you're having the solvents involved and you're having binding solutions. And also it's less energy to do. The negatives, there's more steps. So you have wetting, drying, milling, wetting, drying, milling, wetting, drying, milling, right? There's all those steps, whereas in dry granulation, it's simpler. It also requires more equipment. There's a lot that goes into solvent selection, and then the wetting and drying stages are more time consuming, so it takes longer. So yeah, we kind of talked about how to do it. Important factors, I mean, you have to worry about a lot: the powder size, the solubility, the binding substance, the different types of milling, and then different types of drying ovens. So when you're doing the drying processes, if you over dry it, it will denature some of the chemicals, or some of the reagents, and it might denature the binding solution. In that case, everything will just crumble. So you really need to focus on not just your temperature, but your ramp speed of your temperature. That's very important. Yeah. Okay. So any questions on that? Yes. What's that? Depending on how big the stock is, I'm not sure. Would you know that question? Would you know that answer? How long? Oh, how long would the drug be for wet granulation on average? Right. It take longer. Right. So it depends on the size and then the moisture amount. So I can imagine maybe on some smaller batches, hours to days, I'll say on average. That's about how long these processes take. All right, so now the simplest one, direct granulation. So direct granulation, it's the components are mixed together and then they are compressed. That's it. And they make a pill. Very easy. So it's the cheapest. There's no grinding. There's no wetting. They are mixed. There's no extended mixing. There's no it's only mixed in 1 or 2 steps, not like 10 steps. It's really made for, well, it's really useful for singular drugs that are a larger part of the pill. So for example, vitamin C, or let's say if you take a caffeine pill, or if you have a sugar pill for whatever reason, a zinc and different ions that are just in their salt form, and you can just compress them into a tablet. So the positives, it's cheap, it's fast, not much machinery, it's only 2 steps, and there's no moisture or heat involved in that process. And the negatives, though, it's limited, right? We can only do more pure substances with this, and more complex molecules won't react well to direct granulation. And there's some variation in composition sometimes. If you don't have enough, you don't have any moisture at all. If it dries out, it could create an unblending, meaning everything kind of falls out. So really, the steps, you grind it sometimes. Most of the time, they do grind. You do dry mixing and then tablet compression. That's really it. Yeah, not too difficult there. So I would just say know the difference between all 3. So if I ask a multiple choice question on the final of which process involves choosing a solvent as a binding agent, and you would say, oh, wet granulation. Or so which process involves, or which process does not involve compression? And I would put, or which drug does not involve compression? So I would put in wet granulation of a tablet, dry granulation of a tablet, direct granulation, and then I'd put in like preparation of a biologic. And a biologic doesn't require compression. It's mostly IV for those. So things like that. So here's a summary of what we talked about of advantages and disadvantages of all 3 techniques. So just a summary if you wanted to look for studying purposes. And here are some recent advancements in different techniques that you can look up on your own that are kind of mixtures of the 3 techniques that we learned about. So there's, I spelled this a typo, activated, activated. Moisture activated dry granulation. So there's a bunch of them that are a mixture of the other techniques that are new and evolving. All right. So and those will not be on the exam. So we'll talk about preparing liquids now. So we went from the 3 types of tablet formation, so the granulation. Now we're talking about liquid dosages. So antivenoms, and usually they're liquids. Different types of syrups, IV solutions are liquid based. So yeah, or you could just take like yeah, I mean, really a lot of drugs that are. Where do I have the people? Oh, here we go. So a lot of drugs that are not able to be swallowed by either young children or elders are going to be prepared in a liquid form. And it could be the same drug in a liquid form versus a tablet form. And so I know when I had tonsillitis back in February of 2020, like right before COVID. Yeah, now I'm realizing I'm so glad I got it in February of 2020 rather than a couple months later. I wouldn't have been able to get to a hospital to get surgery because I had to get my tonsillectomy. So if I waited, or let's say I didn't do anything, or let's say I didn't have that problem until March or April, I wouldn't have been able to get to a hospital. So glad it worked out that way for me. But anyway, it was brutal. I couldn't swallow. Like when my tonsils got really swollen, couldn't swallow at all. It would hurt. Really excruciating pain. So I would have had to take liquid. I really had to eat all liquids. I couldn't eat anything solid. It would hurt like hell. So, for example, if people who have diseases or illnesses that impede their swallowing, liquid dosages would be beneficial. All right. So water is the most common solvent, right? Universal solvent. And things you need to keep in mind are pH, temperature, and concentration. So these are very important things because when we're dealing with liquid solutions, we're dealing with concentration. And the concentration strongly determines the effect of the drug. If your concentration is off by 2-fold, you can kill people. So, or you can cause everyone to throw up, right? You don't want to do that. Yes. Yes, usually the water is ultra purified. So it's like we call it HPLC grade. So it's really high end water. So like or molecular biological grade water, same thing. It's really water that it's distilled, it is ultra purified to take out all ions, and it is RNase and DNase free water. So it's really there's nothing. Yeah, reverse osmosis water also. Yes. So yeah. Here's a business idea for anybody that's interested. So in pharmaceuticals, water, that this level of water is really expensive, believe it or not, for water, right? So we are in my company right now, since we're expanding labs, I am trying to order a ultra purification system. The ultra purification system is around like $10,000 to $20,000, which is nothing for the company. But so until that point, I need to order my water from Sigma or Thermo Fisher. For a liter of water, of ultra purified, RNase free, DNase free, reverse osmosis, it's like $45 to $50 for a liter, right? So I'm doing my surface plasmon resonance technique, which is my SPR machine, which requires at least 2 liters every time I run it. So that's a lot of money. So here's the business idea. If you bought or you and a couple of buddies invested in an ultra purifier that it takes tap water and uses UV to actually kill all the bacteria and everything, and then goes through filtration, multiple filtration processes, and reverse osmosis. You make ultra pure water, bottle it, box it, and sell it door to door to pharmaceutical companies, and because there's so many around here, and like send emails first and call, but door to door, and then undercut what they're getting their water at now. But you'll still make a lot of money. So that's a business idea if anybody wants to pursue that. I was thinking about it for that company, but anyway, yes. Yeah. Everybody uses deionized water, but usually for like really high-end techniques, we want to make sure that it's ultra pure. Yeah, but a lot of techniques use that. So I use it in my TR fret assays. I use just regular deionized water. Anyway, what container? So here, I'll just show it. Oh, no, not glass. So it's plastic. Right. The BPA. Yeah. But in this case, so that's over long-term storage. Yes. Right. So what are you asking? Big, oh, sorry. Well, because I mean, like the BPA's are in plastic, and that's what diffuses into our water, which is a carcinogen. That's what comes out of the plastic. But usually that happens over time. Is right? That's okay. Yeah. Okay. Yeah. After class, we'll talk. Okay. All right. So actually, bro. So back to this. Preparing liquid solutions, liquid dosages. So advantages, like I said, patients who can't swallow, or can't swallow pills. So faster absorption rate, and they're more flexible in achieving proper dosages. For example, for pills, let's say each pill is 200 milligrams, but your dosage your perfect dosage would be 300 milligrams. We're gonna take a 1.5 pills. You can crush a pill, definitely. But with water or with liquid solutions, you can just fill up the volume that you need to get that dosage and then take it. So it's the dosages are more flexible. Disadvantages, really, they all have to do with storage. So short shelf life, and they get unstable. Special storage, usually -20 or 4 degrees, and they can be easily infected by bacteria. Yeah. Any questions on that? All right. So, in liquid manufacturing, we need to keep a strong eye out for the excipients, which are the non-active, the protectants. So this includes buffers, antioxidants, preservatives, colorings. There's a lot of different electrolytes, sweeteners, and flavorings. So these are the non-active ingredients that are added to a liquid dosage that are important. Just wanted to show them, because taste is important. Some like what am I thinking of? Robitussin, right? Robitussin tastes terrible, right? We all know that. Why don't they change the taste of it? Anybody know? So kids don't drink it for fun. It tastes bad for a reason. So they maybe at first it tasted like water. A lot of these things do. Or maybe it tasted good, right? But they actually put flavorings in it that made it taste really bitter and really nasty, because they don't want children going in and drinking the whole thing and getting an overdose. So that's why medicine is supposed to taste bad. And then you have like different sweeteners, electrolytes, and stuff like that. So anyway, so here's the workflow of liquid manufacturing, of liquid drug manufacturing, and it's similar to the granulation. Because look, what are you doing? You're taking the raw materials, you're mixing them and measuring them, and then you are mixing them, in this case, with its solvent. That you're not going to end up drying it. You're going to leave it as wet, and then you're going to fill it, package it, and then go to the finished product and do quality assurance. So there's actually less steps than wet granulation, because you're not going through a crystallization, drying, wetting, all those processes. You're only going through mixing it with the solvent and then filling it into containers and then packaging it. So there's 5 different parts of liquid dosage handling, or liquid dosage preparation. And these are not going to be on the exam, but maybe 1 or 2 multiple choice. Nothing crazy like explain all 5, because that's just memorizing stuff. So the tank selection for where you do these reactions in is very important, because whatever tank it is, you don't want the tank to have the inside lining of the tank to actually react with the liquid. That would be really bad, because over time your batch would have a lot of impurities, and also could have metal in it or whatever. So usually Teflon and glass lined tanks are non-reactive with a lot of stuff. So yeah, so that's usually the really this obviously the size needs to be big enough, but really the tank material is the most important thing. Then mixing, you need to have different mixers that are powerful enough and that have, I mean, especially when you're dealing with high viscosity, so like lotions, right? If you're mixing a lotion, you need to have a heavy duty stainless steel mixer that's a lot of power in order to, because lotion is pretty viscous. And then dispersion. So for dispersion, we want to kind of put pressure on different emulsions, and this is not for water soluble, water based liquids. These are more for emulsions and different types of suspensions. So what we're doing here is we are putting pressure and it kind of helps in the mixing and dispersion of the product throughout the emulsion, which is with the API throughout the emulsion. So this is very important, but it's one of the processes. And then we have filtration and clarification. So there's a lot of the QCing, and the filtration is important too. We want to filter out all impurities. And that's really it to this. I mean, there's different filter pads. So that's about it. So, and then transferring and filling. So we want to fill up the batches, whatever batches we get, we want to transfer them into tanks and then eventually, or final tanks, which can then get distributed into the containers that they go in, which goes to the pharmacy or the hospital. Yeah. So yeah. So in this case, if we're doing batch production, new batches should not be started before the previous batches are done, and also emptied and cleaned too. Yeah, and then the bins are important. Piping is important, but yeah. All right, so I mean, don't go crazy with the liquid preparation. I went over it pretty briefly, so we don't want to go too in depth. So the next, unless there's any questions on liquid. Oh, okay, cool. So the next thing, and we're almost done. Or actually, we're almost done. So we have 2 more slides. So biologics. This is more of my realm. So the production of biologics. We're talking about antibodies, proteins, peptides, RNA. They are the best. They are the future of pharmaceuticals. I'll tell you right now. Large molecules are the future of pharmaceuticals. From drug discovery from 1918 whatever to the first drug to alcohol for cuts. In like the 1000 BC, whenever they first used rubbing alcohol or ethanol as a disinfectant. That's a small molecule disinfectant. Those are all small molecules, and 90% of drugs up until this point have been small molecules. So 500 Daltons or less. Now, since our technology is getting a lot better and we're pinpointing diseases more, and we want more on-target effects, and our drug delivery methods are getting better, the focus is shifting to monoclonal antibody treatment for a lot of different things. For COVID, for cancer, for diabetes, for everything, for Alzheimer's. So that's the advantage. The main advantage is the best drug efficacy, the most specific, and it's new and emerging. But the disadvantages are battling right now with the advantages. It's neck and neck. The disadvantages: price, storage. You need cryopreservation for antibodies and proteins. So to give you an example, I ordered a protein from the UK, and it came yesterday. The protein vial was this big. It was a 200 microliters in a 1.5 milliliter Eppendorf tube. Very, very small. This vial was in a bag, pretty big bag. The bag was crumbled up. This bag was put into a giant foam box in the middle with 30 pounds of dry ice. So my boss is like, Patrick, your protein's here. I'm like, I go up to the front. Where is it? There it is. The box is probably like as big as this. So I'm like, I only ordered like 200 microliters. And he's like, yeah, I know. Like it's they went overboard with the dry ice. So I was like, does anybody need dry ice for the next to ship anything? Because so proteins are shipped on dry ice. Dry ice is usually at minus 120 Fahrenheit. No, minus 120 Celsius. Yeah. And then liquid nitrogen's at minus 200 something. But yeah, so dry ice is usually we ship our proteins on. So they need to be cryopreserved, very, very cold, which that costs a lot of money. So imagine the price of taking liquid dosages or tablets and shipping them from the UK to here, dry tablets. It would be like in a normal bubble mailer, right? But in this case, I needed to get protein or a biologic in 30 pounds of dry ice. So the price adds up. And take that on a larger scale, that's millions of dollars of difference. So anyway, that's one of the, and also stability. So proteins and antibodies can easily become disturbed and denatured. That's a big factor. Also, one of the biggest, probably the biggest one, cell culture contamination. You can raise a cell culture for a few weeks on a huge scale, and then you see an impurity in the cell culture, and the cells start behaving weird, and you have contamination. And then you have to throw out the whole batch and start again. So before these, because you're using these cells to purify the proteins, you're using the cells to express the proteins. And you're using the cells to express the antibodies. And you're using the cells to produce the RNA to then take the RNA out and do PCR. So, and also all of these processes and all of these problems make it very, very expensive. Yes. More often than you think. So I've personally never contaminated a cell line that I know of, but you need to be very, very careful in a level 2 biosafety lab, and sometimes level 3, depending on which cell line you're using. But everything is sterile. So they use different tips, they use different pipes, they use a completely different manufacturing plant is for sterile. And that all those regulations, money. So it happens though. It still happens. Yeah. And that's a lot of money. So yeah. So if we want to take a brief look at the workflow, which it's long, but we'll just take a little look at it before we get into some fun stuff. We'll finish on a really fun note, I promise, and you'll learn a lot from the next slide. We'll talk about colors. So master cell bank, then you go to a working cell bank, which means you just have your cell line. Then you centrifuge down your, kind of like you do for Western blot, you recover your proteins or your antibodies. You go through many stages of chromatography. So separation, chromatography is separation. So you are doing chromatography of maybe affinity chromatography, followed by size exclusion chromatography, followed by filtration processes, in order to really, and again, chromatography, more filtration, and then formulation. So really the goal is you're taking your biologic from your cells and you are doing an ultra purification of that product, whether it be through all these different chromatography techniques. Whereas Western blot, we only purify it through an SDS gel, right? That won't cut it on a drug scale. So we want to be more particulate here and cleaner. All right, so any questions on this? Okay, fun stuff now. Color. So I will probably give a couple questions on this because it's super interesting. And I learned a lot about it, actually. So my girlfriend is a cosmetic chemist. She works for Sun Chemical in Tarrytown, New York. And she is in the color and effects department. So she makes, for those of you who are interested in makeup and everything, she makes makeup and like eyeliners and stuff like that. And she actually not just mixes them together, but she actually works on these kind of things, the manufacturing formulations. So it's a really big industry. The cosmetic industry makes more money than the pharmaceutical industry, or makes more profit margin than the pharmaceutical industry. Believe it or not. Yeah. Well, I'll just go into that more. So the reason is because pharmaceutical industry requires much more R&D. Makeups do not need to be FDA approved. There's regulation agencies, but you don't need the FDA. Pharmaceuticals, obviously drugs, you need FDA approval. You need more high-tech instrumentation. which is more money. And then the steps to FDA approval is much more difficult, which means more money is spent without the revenue being returned. But in cosmetics, I feel like my girlfriend talks about a different product that they make just by mixing two old colors together and making a new color. They're like, all right, new product line, new product line, new product line. And there's all these different new makeups that so they're the raw material supplier to like L'Oreal and all these different makeup companies. So they love they market, you've seen the commercials, they kind of market any different new thing that even if it's two old things mixed together with a different name, they just sell everything, and people buy it. So there's a lot more revenue there. But anyway, the importance of color in manufacturing, which I briefly mentioned it the first day of class, but it's really important. So for a lot of different reasons. First, marketing. So there's color associations with different types of pills, right? The blue pill for Viagra. Also, organization. For example, if let's say Bayer has multiple pain relievers for back pain, Bayer back and body, right? And there's different types of those drugs. Let's say one of them is higher strength than the other. They might want to make the higher strength one a darker blue. The lower strength one, or the generic one, they might want to make a lighter blue. So the person taking it realizes that darker probably means more profound effect. That's something that's more psychology and marketing. And also distinguish their own drugs. And also distinguish their drugs from other companies' drugs. So that's why Advil and Tylenol, they're competitors. What color are Advil and Tylenol? Completely different colors. Advil is just like blue, green, tealish. Tylenol is red, orange, or orange. So it's red. Yeah, it's red. It's more red. And then so they're competing colors. They're opposites. So, and then placebo effect, red versus blue, talking about opposites. So if you have a color that's more red, yellow, orange, it's associated with stimulation. Whether it be, not sexual arousal as in the blue pill and Viagra, but stimulation as in faster heart rate, usually like a stimulant. So, for example, 5-hour energy, right? That's technically, it's an energy drink kind of thing, but what color is the packaging? It's like red or orange, right? Because it's associated with stimulating things, right? Blood flow, right? So that's that concept. And then blue and green is usually tranquilizing effects. So a lot of antidepressants are blue or green or white, right? They have to do with kind of relaxing. Beta blockers might be blue or green. So creams and lotions. Did you know the color of a cream or a lotion or a cream impacts the actual efficacy of it? Weird. So the psychology actually has a effective difference, a functional difference. They did a study where they had maybe it was an eczema cream or just like a skin cream, a skin moisturizing cream, right? And they colored the cream white, which is normal. We've seen that all the time, or like a manila color, versus red. Imagine using a red cream on irritated skin. Even if the cream felt the same and cured your irritation, you would feel weird using it, right? It's red. You would think it causes more irritation. That's the psychology they're talking about. That actually, their study showed it had an effect on the amount of relief that it actually gave and the feeling of the patient too. So it had a functional effect and it had a psychological effect, just from the color of the lotion. That's pretty cool. And I heard that, I'm like, no way. So that's pretty cool. Also, antidepressants, usually a lighter color, yellow, usually for like relaxing kind of thing. White is usually for soothing pain. Brightness. So if a color is brighter, usually it means it's more strength and more efficacy, like I mentioned before. And the weirdest one is culture. So the culture, wherever you're from, not Excel culture, but wherever you're from in the world, can actually determine what colors your drugs should be, because everybody in their culture has different connotations for different colors. Whether maybe they're off slightly, but I mean, we don't realize it much because we're all living in New Jersey and it's kind of a melting pot. But other countries realize it a lot when you compare them to each other. So for example, the color blue So blue, what did I say before? It's a tranquilizing effect. It's used for usually pain relief or non-stimulation, just relaxing, right? Blue. It's a calm color. That's why on interviews you're supposed to wear blue, right? Because it's I don't own any shirt that's red. I have like one Under Armour workout shirt because I used to play for a baseball team that was red, but all my professional clothes and even all my workout clothes are either blue, or blue's my favorite color, but or a neutral color. But blue is calming. So you want to give a calming presence when you're giving an interview or interviewing for a job. So, but in Italian men, meaning like Italian, like Italy Italian, like I'm Italian American, but I don't feel this. I don't have this thing because I don't really care about soccer. But you'll see what I mean by that. So they did a study in Italy where they compared the effects of a blue drug. I forgot which blue drug it was. Let's say it's Advil, and compared that the physiological effects of seeing the drug compared to other countries. And they actually saw the efficacy of the drug decreased in Italian men. And they asked them why. Like why? Because they realized their heart rate actually increased when they were seeing and taking this drug, as opposed to it being a tranquilizing effect. And the reason why is because they're Italy's national soccer team, the Blues, they're blue. So what that brings to mind in most Italian men who like soccer is the sport, the adrenaline associated with the sport, and they actually get stimulated from seeing that shade of blue. It's weird. So, but I kind of understand that because I'm a huge baseball fan. If I saw pinstripes, right? Like Yankees are pinstripes. So if I saw the exact pinstripes that the Yankees, I would start thinking about baseball. And then I would think about my own baseball experience, which would get my adrenaline pumping because I want to play. So I kind of get that. But they actually saw it and documented it in a study. I mean, luckily there's no pills that are pinstriped. That'd be weird. But like with solid colors, they actually saw this in Italian men, which is weird. So that's really cool. But like when I'm saying the sedative powers decreased, I'm talking 20%, not 80%, right? This is very minimal effects, but it was discernible to where it was statistically relevant. Another weird one, this is opposite to what you would think, is that anti-malarial drugs in Africa, right? So Africa suffers from malaria a lot, and because they have the bad mosquitoes that give malaria, right? And malaria drugs are prominent, but I mean they're still tough to get because some places in Africa are remote and it's difficult to find those drugs. But anyway, most anti-malarial agents are actually colored yellow. You would think, and so a side effect of malaria is you get a yellowing of the eyes. But normally your eyes, healthy eyes are bright white. So normally, drug colors are marketed to where the color is associated with the intended effect. For example, lotion, you don't want a lotion to be red because red is irritation. You want a lotion to be white. White means no irritation. Or you want it to be skin color, which means no irritation, right? So, but they saw that for some reason, white pills decreased effectiveness. And they still don't know why. They just found this anomaly. But yellow pills increased effectiveness. Maybe because people associated the color yellow with having malaria. So they took the pill and as a malaria, they knew if it doesn't have a label on it, they maybe assumed it was a malaria drug. I don't know. I'm just speculating, but that's what they found. They found that, yes, your question. So it does. So over time it would if it's untreated, right? Okay. Yes. Exactly. So sorry, I wasn't as concise. So if malaria is left untreated, thanks for correcting me. If malaria is left untreated, your eyes may turn yellow. That's one of the side effects. So it's from the liver damage. Right. Exactly. So that was just a weird finding that that their pills are actually yellow. You'd expect the pills to be, yeah, white in that case. But that's something they found in that study. So anyway, that's about it.
Professor, a quick question.
Yes.
Where do they test, if they test this at all, like the color of the drugs and the efficacy? Are those just like meta studies, or do they actually do this during clinical trials?
So I can find the papers. This was on this was a PubMed article.
Okay.
That I read this from, and the study, I'm assuming the study was done in like a health center or like a trial somewhere in like a doctor's office. But it wasn't like in public, like they went on the street and found people and showed them drugs.
Got it.
So I can find the paper. I'm going to put the paper in the slides too, because that's important. Yeah, that's a good question. But it was published in the PubMed article, and there is data in it, so that data could be referred to.
Right, but when drug companies are making these, are they thinking, okay, we're going to do a clinical trial with same formulation, but this one's white, this one's blue, and we're going to see what happens? Or
sometimes yes, not all the time, but that's a really good question. So sometimes yes, if they feel that color will have an effect on efficacy. Some cases you might not. So like whenever you like usually for stronger colors, if you have like a red versus a blue, then you might want to test it. And if it's for something that's a non-life-threatening illness, so if it's heartburn, right? And you take a red pill versus a blue pill, you want to, and let's say it's cheaper. I'll give you an example. Let's say it's cheaper to make the drug red than it is to make it blue. You definitely want to check, because red is not an ideal color for curing heartburn. So if you give them the red drug and give them the blue drug, see if the efficacy is different. If it's the same, then you can save some money and make the red drug. If they respond better to the blue drug, then you have to spend a little bit of money to increase the efficacy to make it a blue drug.